Hi class, and welcome to this week's version of our videos on Mendelian Genetics. Okay, so we need to know a few things before we get started. You may have learned some of this in middle school, you may have not. It might be totally new to you. That's okay, we're going to go through it step by step, and we'll do some activities in class. So the first things you need to know is some vocabulary. So take a minute, write these down. If you have to pause the video, that's fine. And then I'm going to go through these. So what is dominant? Dominant is an allele that only requires one copy to have the trait present. So in a trait like brown hair, you only need one of those alleles. You get one from your mom, one from your dad. Well, you only need one dominant in order to be brown hair. But if you were going to be blonde hair, you need two recessive traits. So let's talk about recessive. That's an allele that requires two copies in order to have the trait present. Now you can have two dominant, two recessive, or a dominant and a recessive. When you have the dominant and recessive, you only need one copy of the dominant to actually have the trait. So you will show the dominant form. So homozygous is having two copies of the same form of an allele. So our example is two little a's or two big A's. Now, the big A's are the dominant and the little a's are the recessive. Then we have heterozygous or hybrid. So you're going to have two different copies. You're going to have a dominant and you're going to have a recessive, but you are going to show the dominant trait. You will not show the recessive trait. An allele is your form of gene. So I've been talking about these alleles already. It's one form of your gene. It's indicated by our letters. So one letter, even though we write genes in the form of two letters, is actually your allele. And then we have our genotype. It's your actual genes. What is your genetic makeup made of? It's indicated by two letters though. So you can have a capital A, lower A, you can have two capital A's or two lower A's. That is, all those different forms are going to be your genotype. And then we have our phenotype. What the trait actually is. So in the example I gave you of brown hair, if you had that brown hair, that would be your phenotype. If you had the blonde hair, that would be your phenotype. What do we actually see? What is the actual trait we're looking for? That is the phenotype. So where does this all come from? Now, we talk a little bit about Gregor Mendel. He was a monk who studied peas in Austria, and he came up with two laws, the law of segregation and the law of independent assortment. Now, Gregor Mendel is very, very important to us because he's one of the first people to have discovered genetics and how we inherit our traits. So what did he study though? Like I said, he studied pea plants, and he studied 7 different traits in these pea plants. He studied seed shape, he studied seed color, flower, pod shape, pod color, the position, and the stem height. Now, as you can see, there's a definite dominant trait in all of these traits, and there's a definite recessive trait. So it is either dominant or recessive when we talk about Mendelian genetics. When we get further into our studies, we'll talk about genes that are traits that need more than one gene, but right now we are only talking about definitive traits. So for Mendel, he crossed peas. He used pea plants to cross because they have a short period of time from cross until they had a new plant. He needed something fairly simple that he could cross very easily, and he could see the reproduction. He used paintbrushes to cross the pollen to the pistil. So we're using the paintbrush to cross that female portion to that male portion. Each pea obtained is a new organism that could be studied. So we have a parental generation, we have the generation after that, and the generation after that. And we can see how the traits are being passed on. So this is very important to what we were doing. So in his work, let's take the flower color for example. And we have what are called true breeding plants. That means it's all homozygous plants. So if it's purple, purple being the dominant form, that's going to be capital P, capital P. So the purple flowers were capital P, and the white flowers were lower p, lower p. So when we cross those two types of flowers, those homozygous types, we're going to get the F1 generation, which is a hybrid generation. The hybrid generation is going to be a capital P, lower p. So what does that look like? So if we have a capital P, and we have a lower p, I'm going to draw a line under this, and another lower p. When we cross those, we're going to get capital P, lower p, capital P, lower p, capital P, lower p, and capital P, lower p. So as you can see, all of these possibilities are only capital P, lower p. So when you cross a true breeding dominant or homozygous dominant with a homozygous recessive, you will get a hybrid, which is a capital P, capital P. So now what we're going to do is we're going to take those capital P, lower p's, and we're going to cross them with a capital P, lower p. And then we're going to see the results. Now, when Mendel bred these flowers, he saw the white flower go away. He didn't think he would ever see it reappear. But with his study on alleles, we now know that that recessive trait can reappear when you cross these different forms. So what we are crossing is two heterozygous, capital P, lower p, by capital P, lower p. And I apologize, I am not that great at writing on my screen, but bear with me, guys. So the line under is the lower. Right here we're going to have a capital, and right here we're going to have a lower. So when we cross this, we get one homozygous dominant. We have a heterozygote, which remember, you only need one copy of the dominant form in order for it to show the dominant form. So we have two hybrids, one here and one here. And then over here, we're going to have two lowercase p's, which are a recessive form, which is going to show the white form of the plant. Okay, so moving forward, Mendel had his Punnett square, what we just went over. So he took that F1 generation, and F1 stands for filial. So we have the parental, and then we have filial 1, filial 2, filial 3, and it goes on from there. So the pollen, or the male, and the pistil, or the female, he combined. So we have capital B, lower b, and capital B, lower b. That's exactly what we just did with p's instead of b's. And as you can see, you have capital B, that homozygous dominant form, which is going to show you the purple. Then you have capital B, lower b, which is your hybrid or heterozygous form, which still shows you that dominant form, because remember, we only need one form of that trait. And then we have lower b, which shows you the white form. And this was very interesting to Mendel. It was very interesting to people at the time, because they thought that you needed to have the traits that you could see. And not necessarily are you seeing this lower b, but it is still present. And if it was to mate with someone or something that has that lower b, it's going to show up in the offspring. Okay, so I want to talk to you a little bit about the law of segregation. This was one of Mendel's laws. The separation of alleles during meiosis. It's 50% chance dominant and 50% chance for recessive for a heterozygote or a hybrid. So when we look at it over here, let's say your dad is heterozygous for a trait like a hitchhiker's thumb. The hitchhiker's thumb means your thumb goes totally back. It is a dominant trait. If your thumb does not go back from that second joint, then you do not have a hitchhiker's thumb. If you can get your thumb forward though, so look at mine. Mine is a hitchhiker's thumb. You see how this goes back from that joint? If your thumb looks like this when you straighten it, you do not have a hitchhiker's thumb. Now, say my mom was heterozygous for this, and she had a capital A, lower a, and when she goes through meiosis. Remember the process of meiosis, we're separating all of those alleles and chromosomes into separate parts, so we have a unique individual sex cell. So there's a 50% probability that her dominant form will go into one egg, and a 50% probability that it'll go into the recessive form will go into another egg. So what is happening here is we always have the same chance to have a dominant or a recessive form. So that's pretty cool. So let's go on to independent assortment. This was the second law that Mendel talked about. And all this states is two different traits do not travel together and do not affect each other. So as we can see here, we have a parent with the genotype capital G, capital G, capital W, and another parent with a lower g, and lower W. Now, we know when we combine these, we only have the chance of having capital G or capital W and lower G or lower W. So when we combine those to the child, we will get capital G, capital W, lower W. Now don't get hung up on the fact that we have two alleles here. We'll explain that later in class when we do dihybrid crosses. But as you can see, we have the chances, when we look at it, at having capital G, capital W, capital G, lower G, capital W, or lower G, lower W. So we have the chances of having all of those in our gametes, which is very interesting for what we are doing. And basically what this says is there's no rhyme or reason to why you get two traits together, you just get those traits together. Okay, so let's go forward a little more. And let's do some sample problems now. So in pea plants, spherical seeds, capital S, are dominant to dentate seeds, lowercase s. In a genetic cross of two plants that are heterozygous for the seed-shaped trait, what fraction of the offspring should have spherical seeds? So right here you can see we have heterozygous. So we have two plants. So from that problem, if you want to go back, go ahead and go back. We can see that we have a capital S and a lower S. And we're going to cross that by a capital S and a lower S. So we're going to draw our Punnett square. And I'm going to use a little help here. We have a square, we have a line, and we have another line. And that's all we need for Punnett square. So we have a capital S here. We have a lower S here. We have a capital S here. We have a lower S here. So we're going to have capital S, capital S. We're going to have capital S, lower S. We're going to have capital S, lower S. We're going to have lower S, lower S. What this means is we have a 1 to 2 to 1 ratio of dominant to dominant recessive to recessive recessive. So we have dominant recessive, we have hybrids, and we have recessive homozygotes. So we have 25% SS, we have 50% capital S, lower S, we have 25% lower S, lower S. So going back to our problem, the question it actually asks us is what fraction of the offspring should have spherical seeds? And the spherical seeds are the dominant seeds. So we had 3 out of 4 being spherical seeds, so your answer would be 75%. Okay, this wraps up our lesson for today. We will have some practice problems that I will email you over the weekend, and they will be due in class on Monday. Have a great weekend, everyone.